Welcome back to this channel. So today we are going to discuss about section B of the sample question paper, and if you haven't gone through the section A, make sure you check that video out. So let's begin, shall we? Now, like I said before, section B comprises of questions which is of 3 marks, right? Okay, so always remember to write your answers in points. Okay, I will keep repeating this again and again that you have to write your answers in points. Alright, so let's discuss the question of section B. How are we supposed to write it? Okay, how can we give out the information that we have in our answer sheet? So, okay, starting with the 7th question in section B, it says that how do normal cells get transformed into cancerous neoplastic cell? Neoplastic cell is nothing but tumor cell, right? So they have asking the question that how a normal cell is getting transformed into a cancerous neoplastic cell. The next part of the question here is elaborate giving 3 examples of inducing agent. Alright, so now we have studied in the chapter Human Health and Disease about the different inducing agents, right? So first part of the answer you have to tell that how this normal cell is getting transformed into a cancerous cell, just by using some physical, chemical or biological inducing agents, right? That is the perfect answer for this first part of this particular question. Now next they are asking give examples of the inducing agent. So you have to give examples for the physical one and for chemical and one from biological. Okay, try to give examples from each one of them. Alright, so physical you can say X-rays, gamma rays, and for chemical you can say the carcinogens from the tobacco smoke which we get, and also for the biological you can mention about the oncogens, right? So let's just see the answer whether they have mentioned everything which I said. Alright, so as you can look here, they have mentioned about normal cell getting transformed to a cancerous neoplastic cell when they are induced by physical, chemical and biological agent. Perfect answer. Next part, they are giving examples of each of this inducing agent. That is for physical agent, they are mentioning about the X-ray and gamma rays, which is nothing but an ionizing radiation. And they are also mentioning about UV rays, which is a non-ionizing radiation. And then the next part they are talking about the chemical agents. What are those? Carcinogens, which is present on the tobacco smoke. Then talking about the biological agent, they are mentioning about the cellular oncogens, proto-oncogens. What does it do? It basically gets activated under certain condition which basically causes cancer. Perfect. You have to mention all of this. That is, you have to mention each 3 inducing agent and examples for each 3 of them. So you will get the perfect 3 marks, right? Okay, moving on to the next question. Okay, so this is the or part of that particular question. So let's just see what does it say. A person is suffering from high-grade fever. High-grade fever is usually about 39 to 40 degrees Celsius. You can say that. Okay, so this person is suffering from a high-grade fever. All right. The question here asked is which symptoms will help to identify if she or he is suffering from typhoid, pneumonia and malaria. Very simple question, a direct question you can say. So in this question they are just asking about what all symptoms are present in a patient who is suffering from typhoid, pneumonia and malaria. You just have to give up the symptoms which you have studied. This symptoms you have studied in the Human Health and Disease chapter, right? So let's just see the answer and see what all symptoms they have mentioned here. So as I said, high-grade fever, typhoid. For typhoid they are mentioning few of the symptoms, that is weakness, stomach pain, constipation, headache and loss of appetite. Next, for pneumonia, they have mentioned fever, chill, cough, headache and everything, alright. For malaria also, there is chills, high grade fever and which is recurring every 3 to 4 days. So you have to mention about the symptoms for each and every one of them. Then you will get the perfect 3 marks, alright. Very simple question, very direct question. Now you can see, you have seen different types of question all through the question paper until now. Alright, so let's just move on and see what other type of questions we can see in this entire question paper. Alright, moving on to the 8th question. So it says recognition of an antigenic protein of a pathogen or exposure to a pathogen occurs during many types of immune responding, including the active immunity and the induced active immunity. Now you all know we in our life stages, you can say, we encounter many pathogens and our body is trying to overcome the effects of those pathogens, releasing some things, alright. So in this question, this condition is given. Basically they are talking about how our body is creating immunity towards certain pathogens, how they are getting encountered with certain pathogens and what are the responses which is being given out in terms of when pathogens are trying to attack our body. Alright, now the question here is specify the type of responses elicited when a human being get encountered by a pathogen. So here you have to mention about the various types of responses which a human being, you and me basically, gets encountered when they are encountered by a pathogen. What sort of responses do we give when a pathogen is being encountered? Alright, so when we are encountering for the first time any sort of pathogen, the intensity of our response will be very low, right? But again, if that same pathogen is being encountered again, then the intensity of the response will be high, alright? Again, so when a high response is given out, there are this special type of cells which is being produced. We have studied about this, the special cells that are the lymphocyte cells. Remember the T cell and the B cell. So T cell and the B cell are produced during this high intensity response action. Alright, so what is this? You in this question, as it's of 3 marks, it will be good if you mention about the functions of T cells and B cells. So what does B cells do? They basically produces a protein. This protein is called as antibodies, right? Remember antibodies. So B cell produces antibodies, right? B cell is producing antibodies. What does the T cell do? T cell is doing nothing, but it's just helping the B cell to produce those armies of cells, alright? Okay, so you have to talk about the first response, that is the response will be low. The second response, that is the encounter with the same kind of pathogen, that time the response will be high grade response, that is of a high intensity response will be there. Then they are producing B cells and T cells because of this high grade response, and then just mention about the functions of T cells and B cells, that is B cell is producing an army of proteins. What is it called? Antibodies, alright. So they are producing this antibodies in response of those high grade pathogen response, and then T cell is basically helping the B cell to produce this army of cells. Alright, let's see the answer, whether they have mentioned what and all I have talked about. Alright, so they are talking about how from the first encounter it produces a response which is of low intensity because our body is retaining the memory of the first encounter. Perfect answer. Okay, then they are talking about how the subsequent encounter, what does it our body do, basically have a highly intensified response, and it is carried out with the help of two special types of lymphocytes, B lymphocyte and T lymphocyte. Perfect. And then you have to just talk about the function of B lymphocyte and T lymphocyte, that is response to pathogen in our body. It produces an army of proteins, and these proteins are called as antibodies. What does T cell do? It helps the B cell to produce them. Alright, perfect answer. Let's move on to the next question, that is the 9th question. We are halfway through our paper, so just keep up your spirit and try to learn as much as you can in this video. Alright, okay, moving on. In a pathology lab, a series of steps were undertaken for finding the gene of interest. So in a lab, a series of steps were taken to find what? A gene of interest. Alright, perfect. The next part of the question is talking that describe the steps or make a flow chart showing the process of amplification of this gene of interest. So you have to talk about how a gene of interest can be found. Okay, so how you can amplify the gene of interest. So using the PCR technique, that is the polymerase chain reaction, the 3 steps of polymerase chain reaction. What were the 3 steps? First, it undergoes denaturation. You have to explain about denaturation. Second step is annealing. Alright, second step is annealing. You have to talk about annealing. And the third step is extension of that particular strand. Right, so if you are mentioning about these 3 steps and talking about it briefly, just discussing about what is happening here, you can get the 3 marks. Or you have to show the flow diagram which is present in our NCERT book of polymerase chain reaction. So try to learn those diagram here. Alright, so let's just see the answer. Okay. So the 3 processes, that is the denaturation. What happens in denaturation? You have studied. That is, the DNA strand is basically treated with a temperature of 94 degrees Celsius. That is, basically it is being heated here. What happens? The strands are separated. Next step, annealing. Here they are using a primer to anneal the complementary strand. Alright. Next step is extension. They are using the DNA polymerase. What does it do? It basically facilitates the extension of the strand. And what happens? A gene of interest is being amplified. Very simple, right? Very simple. Alright, so just see the flow chart here. So like I said, you can talk about denaturation and you can mention annealing and also extraction. So while drawing the diagram, make sure that you should label the diagram, alright? Like I said, it's very important to label the diagram or you're not gonna get any marks. So especially in this DNA diagram, try to mention the polarities also, that is 5', 3', okay? So that is the diagram, you can draw that for the PCR technique, okay? So let's move on to the next question. Let's see what is it talking about. So 10th question is having 3 parts. And the question is for 3 marks. So it's obvious you have to give 1 mark answer for all of these 3 parts, right? Understood? Okay, let's see the first part, that is the A part. What is it talking about, right? So here they're talking about the evil quartered. What was evil quartered? Evil quartered is the nickname which is given for the causes of biodiversity loss. Causes for biodiversity losses. It's a nickname which is given for evil quarter. You have studied in the chapter biodiversity, right? you can see so here the question talking is the evil quarter describes the rate of species extension due to human activities. So what are the different causes of biodiversity loss? First is fragmentation, alright. Then they are talking about overexploitation, and then they have talked about the alien species invasion, and then they are talking about co-extinction, right. So from that 4 causes, here the question is asked from fragmentation. Okay, so the question here is explain how the population of organism is affected by fragmentation of habitat. You have to answer about how it is affecting when a habitat is being fragmented. Okay, so let's just consider that here is a population. Okay, there is a population going on. Fine. Okay, let's say A, B and C. These 3 populations are staying in a particular habitat, alright. Now they are being fragmented. A is staying here, B is staying here, C is staying here. So what is happening here? So when there was A, B and C together in a big field, they are getting resources, food, and I know there will be a concept of competition and survival of the fittest here, but they are getting food out of it. Okay, so they are getting everything, they were getting unlimited resources. But as soon as they are fragmented, what happens? Those resources becomes limited and their survival becomes hard. So what is happening? Eventually their population will decline. So you just have to explain about this, that if they are getting fragmented, then they won't get enough of resources. So eventually what happens? The population will decline. Okay, perfect. You have to answer that. Okay, now moving on to the second part. So second part is talking about introduction of alien species has led to environmental changes and decline of finding indigenous species. Give any one example of how it is affecting the indigenous species. You have studied about water hyacinth, right? Parthenium. What does it do? It basically, when it is introduced into our water, okay, when it is introduced into our water, it basically consumes the oxygen which is present in the water. And what happens to the species who are staying in that particular water? That is, the fishes who are staying in that particular water will not get oxygen only, right? So what will happen? The population will decline, that is, the fish population will decline. So that is how, if you introduce an alien species, for example, that is water hyacinth, if you are introducing that alien species to a particular area, that is into a water, the species which is present in the water will get affected. Okay, so you can explain, very perfect example for this question is water hyacinth. Okay, you can explain that. Then the third part of the question here is, could the extinction of stellar sea cow and passenger pigeon be saved by men? Okay, so they are talking about stellar sea cow and passenger pigeon, and the question is, can they be saved by men? Now, you all know men is greedy, okay, and we have many needs, right? So if we just reduce those greediness and reduce our needs, then definitely the resources for both of them will be ample enough for their survival, right? So if we have a sustainable harvesting kind of environment, then they will eventually survive, right? So you have to just explain about how if you can reduce our, if men can reduce their greediness, or if they can reduce their neediness, then of course we can save any, species in this earth, right? Okay, let's see the answer. So, okay, so for the first part, they're talking about how small fragments are done. due to which various activities, especially for mammals and birds, and also for the migratory animals, they are also getting very bad affected because their population will decline because of their resources is limited. So you have to explain that. Perfect. For the B, that is example of how introducing an alien species has affected. There are various examples. You can talk about Nile perch, and also you can talk about water hyacinth, that is Parthenonium, which I have given you the example of, and also you can talk about African catfish. Whatever feels good for you, mention any one of them, alright? But study all of them so that what if 2 marks question arises from this topic, alright? Okay, then the third question, that is, humans have overexploited the natural resource. Why? Because we were very greedy. Okay, then our needs. Okay, we were very greedy, that's why we have overexploited the natural resources, so which lead to the extinction of half of the species of the animals. All right, so how can we improve that? By using a sustainable harvesting, right? So by sustainable harvesting we can reduce that problem. Fine, if you explain all of this, you will get your 3 marks, right? Okay, let's move on to the next question, that is the 11th question. Okay, so here an image is given. Okay, let's see what image is this image, right? Very colorful image. Okay, so again this question is for 2 parts. Okay, let's see what the question is asking. The image shown below is that of a sacred grove. Okay, so this image is of a sacred grove found in India. Perfect. Okay, now the question here is, explain how has human involvement helped in the preservation of this biodiversity rich region. So they are asking how us humans have helped these sacred groves to preserve, like how we humans have preserved these kind of sacred groves of areas. So first of all, when we are talking about India, India is rich in culture and we have various religious belief, right? So in various religious belief, we always have make sure to conserve those religious belief and protect all of this religious belief. So some of these areas or some of the land, we can say the sacred groves are the lands which is being protected by us humans in terms because we are having some sort of religious or cultural beliefs on that particular area. So that is how that area is being protected by us humans. So what happens when we are protecting that areas, various floras and faunas are also protected there. So that is how us humans are acting as the people who preserve our environment. Okay, so you have to explain about how we are having this sacred groves, because we have various religious and cultural beliefs which emphasizes us to protect all these areas. Perfect. Now the next part of the question here is asking about value of Z, that is regression coefficient. Now we have studied about the species area relationship. In the species area relationship, there is a formula which is given by Alexander Van Holt. What was it? Z equals to S equals to Z, CAZ, right? What is this Z? The regression coefficient. Alright, so in this question they are asking value of Z is considered for measuring the species richness of area. Perfect. So they are asking, okay, so they have mentioned about species richness area and they are asking if the value of Z is 0.1 for area A and 0.15 for area B, which area will have a higher species richness and a steeper slope. Now considering a graph, there is a slope. Here you can mention about species richness. Okay, species richness. Now as here there is more steeper slope you can see the species richness will increase. So that is your answer right there. What happens as this slope, that is the steeper the slope, the species richness is also increasing accordingly. Alright, so in this 0.7, that is area A will be having a higher species richness. How? That is, this is the regression coefficient. Consider this as the regression coefficient, that is Z. So if you can see 0.17 is mentioned here and 0.15 is mentioned here, you can see as the slope is going above, the species richness is increasing. So that is how 0.7 will have the steeper slope and high species richness. So that's it, you have to explain that. Let's see the answer. Alright, for the first part, they're talking about India's history of religious and cultural tradition as emphasized for the protection of nature. Okay, in many culture, tracts of forest are set aside, all the trees and wildlife within are venerated and given protection. And also talk about sacred groves. Okay, sacred groves in many states are the last refuges for the large number of rare and threatened species. Okay, their conservation would not be possible without the active participation of us humans or the local people who are staying around that area. Now try to give example. Okay, whenever it's possible, try to give example in your paper. Okay, it will be a plus point for your answer also, so always try to give an example. Okay, next part of the question, when z equals to 0.7, z equals to 0.15 for area B, area A will have a steeper slope and high species richness. Perfect answer. Okay, moving on to the 12th question. So here an image is provided to you, right? What is this image? It's that of a gel electrophoresis. You have studied in the chapter biotechnology and its principle about gel electrophoresis, right? So in gel electrophoresis, DNA are being loaded here. You can see that DNAs are loaded here and there is a ladder sequence, which is, that is the DNA ladder sequence is provided to you here. And then you can see that the sequence is being separated, that is fragments of DNA are seen in this image, right? Now in the question, let's see what is the question about. So if the ladder represents the sequence length up to 3000 base pair, the first part of the question is which one of the bands are corresponding to 2500 base pair and to the 100 base pair. So they are just asking which of these 1, 2, 3 or 4, which of these sequences corresponding to 2500 base pair and to 100 base pair. Now you all know that the heavier sequence, that is the heavier sequence will move less, right, in the DNA gel electrophoresis, and the shorter sequence or the lighter sequence move the farthest. So you have to just put on this concept to understand which one is that of a 2500 base pair and which one belongs to 100 base pair. Now you can see the third one is moving the less, right? So it's obvious that it's the heavier sequence and it's that of a 2500 base pair. Clear? Now you can see the fourth one is the one which is traveling the farthest, right? Which means it's a shorter sequence and it's of 100 base pairs, right? Clear about the first part? Now moving on to the second part, they are asking explain the basis of this kind of separation and also mention the significance of this process. Now let's concentrate on the first part of this second part of question, that is the basis of this kind of reaction. How is this fragments getting separated? So first you can explain related to the size itself, that is the heavier sequence moves the less and the shorter sequence moves the farthest. Okay, you can explain with the size. And second one you can explain with the concept of charge. That is, you all know DNA is a negative charge, right? DNA is a having negative charge, right? So it's obvious that it will move towards the positive side. That is, the DNA is negative, so it will move towards the positive side. That is the cathode. From anode to cathode, the DNA will move. Okay, so just the simple trick to remember which one is anode and which one is cathode, just try to remember the concept of panic. That is, the positive one is that for anode and the negative one is for cathode, right? Clear? So explain with the help of size and with the charge this basis of separation. Fine. Now the second part of the question is mention the significance of this process. Okay, so why do we separate the DNA fragments? Basically to get a purified DNA so as to make a recombinant DNA, right? So that you can put it into a cloning vector. So just explain that concept and you are sorted for this question. Now let's see the answer. As I said, band 3 belongs to the heavier sequence, that is of 2500 base pair, and the band 4 is that of a lighter sequence, that is 100 base pair. And the basis of this kind of reaction, that is due to charge, that is DNA is negative, so it will move to the positive side. And according to the size, that is the shorter molecule will move the farthest and the lighter molecule will move the farthest, and the one which travels the least distance is the heavier molecule, right? So, and the second part is the significance of this separation is basically to purify the DNA from the fragment for its construction in the cloning vector, right? Perfect answer. Clear? Let's move on. So we have finished our section B. So as you have seen in section B, they have talked about the chapter biodiversity and they were talking about biotechnology, and the questions are from human health and diseases. So if you are concentrating on these chapters, it will be great for you.